Hi, this is Professor Hinanit Koltai from Volcani Center, Israel, and I'm very glad to participate in Professionally Cannabis.
Where do they go? The smoke rings I blow each night. Oh,
Hello again, listener, and welcome back to Professionally Cannabis. I'm Johnny Weiser, and with me is my co-host, Oscar Houseman. Oscar, we had a really interesting and exciting guest for this week's episode. Would you like to tell the listeners who we had on the show?
Yes, well, I'd love to introduce Professor Hinanit Koltai, who you made me pronounce the name of with a little twinkle in your eye. But at the Volcani Institute out of Israel, I don't want to get ahead of myself, but this is fascinating stuff. We sometimes talk about science on the podcast. We're generally business focused, but this is talking to a leader in the scientific field about how their research, their drug development gets into the hands, or in this case is on the skins of patients. I think it's a really exciting one, really interesting to see just how those years of research tick along until something that can eventually cure cancer.
Absolutely, and not to give too much away, we touch upon how products are going into actual clinical trials in Israel, in addition to new studies that are involving tissue printing. Now, hopefully that's enough to whet the appetite. Listeners, enjoy.
where do they go? The smoke rings
It gives me
I
great
blow
pleasure to welcome to the pod Professor Hinanit Koltai, head of the Cannabis Lab at the Volcani Center, a governmental agricultural research facility in Israel. Spearheading amazing cannabinoid studies, we're about to hear about some of the industry's leading research being undertaken using this amazing plant. Hinanit, welcome to Professionally Cannabis.
Hi, good afternoon.
Good afternoon. Glad to have you on the show, Hinanit. So, we like to lead with a question to all our guests of what led you to the plant?
So, I'm a scientist in Volcani Center for close to 19 years now. And at the first 14 years of my career as a scientist here in Volcani, I spent on plant hormones. I discovered new activities, new to science, regarding the effect of plant hormones on root development, and it was interesting. And I got the scientific papers and international recognition and the highest academic degree here in Volcani. But something was missing for me. And this is the ability to lead to development of products for the benefit of humanity, just like that, medical products preferably. So I switched after 14 years of working on plant hormones. I switched with my lab into research in medicinal plants. We started to work on several medicinal plants from the Israeli desert, being used, for example, by nomadic tribes. But then the interest in cannabis started to rise. Since then, 5-6 years ago, we are working on medical cannabis. Each day is a new excitement, new revelation. All of us, I think, are highly excited about the opportunity that we have to work on this plant.
And would you be able to give our listeners a little bit of an overview about the Volcani Center?
Yes, so Volcani is a governmental institute which is focused on agricultural studies. We have in Volcani several institutes like plant protection and animal studies. I am part of Plant Sciences Institute, and in my lab, as I said, we have research, which is basically a mixture of basic science and applied science. I will talk about that more in a few minutes, I suppose.
Yes, I'm sure you will. So I think it seems like there's been a thread running through your whole career. Are you using those early nomadic plant studies and plant hormones in your current cannabis research? Is it all accumulative? Are you at the peak of your plant science game?
Yeah, I think it does. I think that being privileged, I would say, in working so many years on these subjects, throughout the years, I gain insight into biology, plant biology and, moreover, the interface between plant biology and preclinical studies. Gaining this insight allows us, my lab and myself, to develop, to get better more and more, and to be able to answer more precisely questions that we target about mechanisms and the ability to translate our studies into products.
Excellent. So, I think we'll go into those products towards the middle of the podcast, but I'd love to know, what's your general approach? I guess you must have a shared approach amongst both the basic science and the applied science you do in your lab.
As for cannabis, just as I said, 5-6 years ago, coming into this field, I asked myself, as a scientist, what are the gaps in knowledge at that time? And what I discovered is that there are quite a number of studies that are focused on single molecules, either purified from the plant or synthetic molecules. This is, for example, the studies of Professor Meshulam and his co-workers. And then there are quite a number of studies, and especially anecdotal data, about the usage of the whole flower. So what I discovered is that there are gaps in knowledge. And the question to be asked is, what compounds and what composition of compounds from cannabis are the ones that are active for different medical indications? One, in cannabis, you have hundreds of different compounds and dozens of compounds within each and every strain. And talking or using cannabis products only based on THC versus CBD is not informative. This is not how you should get your medicine, based on one compound or two compounds out of dozens of other compounds that are present in a certain cannabis-based product. So that's one. Two, we hypothesized, indeed it seems, that, and it's also reasonable, that you should not use the same medicine to treat different medical indications. Why should you treat inflammatory bowel diseases just as you treat anxiety? With the same flour or oil or joint? There's no real reason to that. And therefore, the question that we ask about compounds and compositions is specific to different medical indications. And for each and every medical indication we are working on, we are asking these questions. What compounds, what composition are the optimal ones for the treatment of patients?
And with regards to studies that are pertaining to the research of patients, I'd love to hear some examples of studies that you and your team have been working on, and ultimately how they might later be applied in, or perhaps already currently are being applied in, medicine and treatment.
I can give two examples, I suppose. One is we have discovered a certain composition of phytocannabinoids, which is the optimal one for treatment of skin cancer. What we did is we work with hospitals in Israel, and in this case, it is Beilinson Hospital in Israel with the Department of Dermatology. And it turned out that patients came to the doctors and told them that when they put cannabis on the skin for a very specific disease, which is cutaneous T-cell lymphoma, they have improvement in, at least in the symptoms of this disease. And this is sometimes with patients that no other solutions could help them in their disease. This disease is based on the accumulation of malignant T-cells in patches in the skin. And this is the first phase of the disease, mucosis fungoides. In the second and more severe stage of the disease, these malignant T-cells are penetrating, filtrating into the blood. And then these patients suffer from Sézary syndrome with usually unfavorable prognosis. So with our partners, the doctors, we asked, how come cannabis is helpful, at least for the symptoms in these patients? And as a matter of fact, is cannabis really helpful? And if so, what compounds are the active ones? We did more than 2 years research, and at the end, we came with a certain composition of phytocannabinoids, which is much better even than using the crude extract for actually killing these cells. We've shown that in vitro, which means in cell cultures in the lab. We've shown that ex vivo on cells of patients, which was very exciting to see that we can kill these cells also in blood samples taken from Sézary syndrome patients. From that, first of all, discovering the actual composition of phytocannabinoids, this is serving now as a base for drug development. In parallel, we identified a very interesting phenomena there, and this is synergy. What we found out that once we combine in a certain combination, at certain ratio and concentrations, these certain phytocannabinoids, we can get effect which is 40 times better. This means we validated that using mathematical models, that indeed we have a true synergy between the different compounds. And now, if you just think a little bit about what patients report and about the entourage effect being reported by patients and identified previously, some years ago, by Professor Meshulam and co-workers, this is synergy. Synergy between phytocannabinoids. So, one, we identified synergies that might very well be the actual entourage effect reported by patients, one. And two, we have set a base for new development of new pharmaceutical products from cannabis. These formulations of phytocannabinoids are API, active plant ingredients, that are specific for a certain medical indication.
Sorry, I just wanted to hear more about the pharmaceutical side of things, but I was also interested, while we're talking about the entourage effect, are these the major cannabinoids that we know, or these minor cannabinoids? And how many do you find are needed for this indication? Is it 3? Is it 30?
In this specific case, we are talking about 4 phytocannabinoids that are working together and indeed giving this synergistic interaction and substantial cytotoxic effect on the malignant T cells. In other cases, we see other compositions. Some of them are simpler, contains, for example, just 2 phytocannabinoids. Others are more complex and contain phytocannabinoids and terpenes as well. What is really interesting for us, in parallel to developing the pharmaceutical products, is the question, what is this synergy? How can it be that cells exposed to one component and the other, and then both components, lead to response which is not twofold, but rather 40-fold in this case? To answer that, we look into gene expression, for example. We go deep into the mechanism, into the mode of action, at least partial mode of action, into identification of receptors, for example, that are involved in a cell's response to these API formulation. And just to finish my answer, you asked me if these are the major phytocannabinoids. Well, as we see it, we do not have any major or minor. All phytocannabinoids are being examined equally. And by that, we are indeed able, I believe, to extract activity that sometimes are being lost if you look only on THC or CBD, or use products based only on those definitions.
Now, we've spoken about sort of unique compounds of certain phytocannabinoids, but something I was keen to know from you, and actually references a previous podcast that we've done within a series, is that how important are the particular genetics and strains of the plants in which these cannabinoids are derived out of?
For us, strains are merely collection of molecules. And if you ask me about strains, or what strains should be used, or what strains are the best ones, My answer is different. My answer is referring to molecules. And by that, by referring to molecules and to certain composition or compositions of molecules for certain activity, then I'm not that worried about strains or about even growth conditions, because for me, I do not care really how you compose the certain product. Basically, you can compose the certain product from different strains. Just take the right molecules and put them together. So you can blend and you can take fractions and blend between fractions from different strains. Once you are working towards certain API formulation, then you can compose that from different strains, different growth conditions even. By that, you're not that dependent on this strain or the other. And as a matter of fact, this is something that patients are suffering from. The lack of knowledge of what strain to use, and then the lack of standardization of cannabis-based products. Once you know what you need in there, once you know what the API is, then you can have a product which is not in particular or necessarily strain specific, one. And two, it can be and should be standardized based on the API formulation. I think by that we touch one of the major benefits of this API-based approach. You open the black box of cannabis. Look what you have inside, choose the right molecules to work with, and by that, you lead to new products that are effective, efficient, and standardized.
So let's say you've got your APIs, you've got 500 APIs available, you've identified what sort of mixtures to use them for what indication. How do you take it out of the lab and into a patient's bloodstream? What's the process?
So, even from the beginning, we start and work with methodologies that are being recognized by the industry and can be translated into products. So, in many cases, we work with commercial strains, as many as possible, to have a variety of molecules. But then these are already commercial strains that can be the raw material for products. That's one. Two is we work in methodologies and with equipment which can be quite easily upscaled by the industry. As a matter of fact, just in this example of cutaneous T-cell lymphoma, we were able, working on the one hand with physicians, with medical doctors, and on the other with the industry, we were able to translate the results that we have gained here in the lab to actual commercial products. And this is even using different strains and commercial methodologies. Once we kept the API, we kept the composition to be the same, we were able to promote our research result into commercial products. Another important aspect of this translation is working with formulators. As for now, in this case of CTCL, cutaneous T-cell lymphoma, we are working with formulators, with Nexstar company in this case, and they are formulating. They, first of all, produced the API, as I said, in their methods, their hands, from strains, commercial strains. And what they do now is they formulate these API that they themselves produced, and we assured their activity. They formulate that into creams to start with. These medical creams will go now into clinical trials in Beilinson Hospital. But one point to consider is that once you have the API, you can take and put them into different drug delivery devices or methods or means. In some cases, you may want to use them systemically, and then maybe by vapors or nasal sprays, etc. In some of the cases, like the example that I talked about, you want to target these products, for example, to the skin. By that, formulators are able to further develop these products using different drug delivery.
So does that mean you do have a product that's now being used in the hospital, or it's just on the cusp?
We have a product that is going now to clinical trials. Since we are talking about a cancerous disease, we have to go through clinical trials to be able to, saying on some product that is anti-cancer, you have to go through clinical trials. I would not be able to stand and say this is an anti-cancerous product unless I show that in clinical trials. So this is the stage we are at. We have the products. We are going now to clinical trials in Beilinson Hospital and possibly in other medical centers to prove that indeed this is helpful, I hope, in patients.
So this must be truly innovative then, because I think there's only Sativex that's done clinical trials with 2 cannabinoids, and does this have 4 cannabinoids? So that means there must be 2 that have not been trialed anywhere.
That's right. Yes.
What would be your estimate of how long in time it will take until it goes from the trials that are starting now to being fully rolled out as a pharmaceutical product that can be administered to patients outside of trials?
I believe that the answer to your question depends on the results of the clinical trial. If they would be successful, I believe and hope that within few months we will be able to start having a product in the market. But if these products would not be efficient or effective enough in treating this disease, then it will go back to the lab and we will try to improve and perhaps start from the beginning. Who knows?
So let's talk about some other products or other research, because we've done quite a good journey with this one, I feel, talking about the initial research, taking it all the way to how it will appear to products. But that's just one formulation, dermatological, and I suppose this is the applied science start of your business or research. What else have you got cooking up there in the Volcani kitchen?
Being a research lab, we are asking additional questions, pushing, I would say, in some way, the boundaries of knowledge for cannabis. For example, one question that we asked is whether terpenes are important at all for medical activity, using cannabis, of course. Some say that the terpenes are not important and not part of the entourage. So we examined that. We developed methods to isolate terpenes and then looked for phytocannabinoids activities with and without terpenes. What we could see is definitely, and published that as scientific paper, definitely terpenes are important for phytocannabinoids activity. So if we think on the entourage, we have the intra-entourage within the phytocannabinoids themselves, and then we have inter-entourage between phytocannabinoids and terpenes. So this is one of the questions that we ask and try to answer. Another very interesting question that we think of is a question that, is being raised many times by students or audience in my lectures. And this question is, why is cannabis so rich and full of compounds? The plant is actually slaved to producing these different compounds. And the question is, why does it do so? Why do we have so many different compounds in such large quantity? Some say that this is because this is part of plant protection. However, cannabis has quite a number of pests, and this seems not to be, at least not the whole answer, all of it. What we believe as scientists, and again, another paper, which is actually under review now, is that cannabis is so rich in compounds because of humankind. We are dealing with this plant, using it for different purposes, medical purposes, but also for recreational use and for ritual use. We're using it for thousands of years. During this usage, we changed plant properties in such a way that we chose individuals, plants, that are rich with phytocannabinoids, rich with terpenes. And doing this selection, breeding and selection throughout the years, we came out with plants that are highly rich in these secondary compounds.
I know from a previous conversation that we've had that one of the projects that you're currently working on is focused around tissue printing. Would you be able to give us a bit of insight into the work you're doing here?
Yes. So we are working now, as I said, for years on human cells, and in collaboration with different hospitals in Israel, also on biopsies of tissues that we receive from willing patients, of course, from different hospitals. Just as an example, for quite a long period, we received biopsies from colon tissue of patients that suffer from inflammatory bowel diseases. And they examined the activity of cannabis directly on the tissue. This, by the way, resulted with a very nice project, again, a project in which new cannabis-based suppositories are being developed for the treatment of for IBD, inflammatory bowel diseases. But at a certain point, we felt that we need to upgrade our ability to examine human tissue, not by using just a rather limited number of biopsies, but being able to ask more deep, perhaps sophisticated questions as for the activity of cannabis compounds on the human body. We upgraded our abilities to ask the question what cannabis compounds, what molecules are active, and how these molecules are active in terms of mode of action, of identification of targets. And we did that by developing in my lab abilities to bioprint human tissues. What we do now is we print tumors, human tumors, from different parts of the body. For example, we print colorectal tumors and even brain tumors. And on these 3D structures, on this printed tissue, we are able now to ask more deeper and more sophisticated questions about the activity of cannabis. By using these printed tissues, we are able to better imitate the disease, the medical condition, and perhaps even patients' response to the different treatments. So we are printing, as I said, tumors. We are starting now to print healthy tissues, to ask additional questions, for example, on the activity of cannabis compounds on inflammation in normal tissues, for example.
And with regards to the application of cannabinoids and other compounds on these manufactured tissues, is it like for like in terms of how those printed tumors and cancerous cells would respond the same way as organically developed cancers?
To verify the resemblance, I would say, between the 3D printed tissue and the human tissue or patient's response to this, and we'll need clinical trials. Only clinical trials would verify this resemblance between the 3D structures that we bioprint and the human body. However, these 3D structures, this bioprinted tissue, allows us to do something very important, and this is to screen in a high throughput manner for different combination of compounds, of cannabis compounds. Think about that. When you have hundreds of different compounds in cannabis, you have many, many, many combinations. And the printed tissue allows us to screen for these many different combinations and to come and to go to clinical trials with only the successful ones.
Amazing stuff. Well, we can't wait to see how things develop on that front. I think we probably have time for one more question or one more bundle of questions. For listeners of ours who might be coming from the more business side of the industry, who've been hearing sort of the amazing research that you've been doing and perhaps would be interested in interacting with you, your team, and the work you're doing at the Volcani Center, how would something like that come to pass? And what type of entrepreneurs / research partners would you and your team want to and or look to work with?
We are looking for collaborations with commercial companies. Commercial companies are those that collaborate with us, of course, are serving as the applicative arm, you may say, of our activity. What we do in my lab is research only. We are not able to actually develop products. Commercial companies that are our partners in this exciting process, I think, of medical cannabis, are the ones to develop the products. So first of all, I'm looking for this kind of collaborations with commercial companies for the development of new cannabis-based products. Just to say that I think that this will be the next generation of cannabis products. And as I see it in the future, these products will be the ones to be used for medical treatment. Other collaborators that we are very interested in collaborating with are medical doctors, physicians from different hospitals from different parts of the world. This is because this kind of collaboration promotes our ability to build bioassays, biological assays in the lab that are able to represent the disease the best we can.
Well, this is all incredibly fascinating stuff, and I can speak for both me and Oscar to say that we absolutely cannot wait to have you back on the podcast and hear about developments across all of these studies and projects. Hinanit, it's been a pleasure having you join us on Professionally Cannabis.
Thank you so much. Thank you. It was my pleasure.
Where do they go? The smoke rings I blow.
Well, that conversation certainly raised my IQ a few percentage points. Listeners, if you enjoyed this content, comment and let us know. But otherwise, we just like the typical subscribes, comments, shares, all the things that help the podcast grow. And see you next week.
Hello, please take me above. Take me with you.